we ready? Are we ready for transcription? No. Questions? Okay, we're going to start with watching a cheesy video, because you know how we love this guy. For the overview of transcription, right, what's transcription? Going from DNA to RNA, DNA template to an RNA nucleic acid. All right, let's see if we can hear him. No. Nice buzzing sound.
Transcription is the process by which RNA is assembled from a DNA template.
Yeah. Sorry. Crap.
Transcription is the process by which RNA is assembled from a DNA template. The two strands of the DNA double helix are held together by bonds between complementary base pairs. DNA encodes genetic information in the sequence of bases along one strand. The portion of the DNA molecule that is a single gene or coding region is bounded by termination and promoter sites. A molecule of RNA polymerase binds to the promoter site. It moves along the DNA, separating the two strands of the double helix. Each now unpaired base will bind to a nucleotide in the vicinity which has the appropriate complementary base. In the synthesis of RNA, uracil is the nucleotide complementary to adenine. This process ceases when RNA polymerase reaches the termination site. The DNA strands bind to one another once again as the new RNA molecule moves away. This RNA is a copy or transcript of the message contained in the gene.
Okay, that was a gross oversimplification of this process, with lots of things that are kind of wrong. But it gives you the basic idea. Okay, so as I'm going through the lecture, I'll try to point out where things were grossly wrong. But it's the simplest version of what transcription is, right? Taking DNA as our template, and we notice only one strand was used as the template strand, right? Because RNA is always single-stranded. That's what we learned in chapter 4. DNA is always double-stranded, except transiently during process. And then, just like in DNA replication, single-stranded binding proteins really need to keep that other strand away for the most part, because it's going to want to jump right back down. But polymerase, kind of as soon as polymerase gets through, it closes right behind it. Right? They showed it staying open a lot longer than it should have, but that's just to make sure it was clear where the RNA was. And as soon as there was enough RNA built, it automatically came off, right? So it's not like transcription happens and it sticks for the whole 10,000 base pairs, and then the whole thing falls off, right? As soon as it gets a few base pairs in, then the DNA-RNA hybrid comes off, and the DNA comes back together, essentially right behind the polymerase, okay? And what else did we notice about the beginning of, yeah, sorry. Why don't, which ones? Right, why it comes off? There's proteins that break it and make it come off. Yeah, it wouldn't normally just come apart. Yeah, so there's proteins involved. So when they say RNA polymerase, it's actually a complex, and so especially in eukaryotes, there's hundreds and hundreds of proteins in there. One of them you could think about on the butt end of that complex is breaking those, and then the two DNAs come right back together. Yeah, because otherwise they wouldn't just, an RNA-DNA hybridization is as strong as a DNA-DNA. right? What's the only difference between a DNA and a DNA-RNA complex? Yeah, the U's that show up in RNA instead of a T. But if you go back to chapter whatever, 4, and look at an A and a U hydrogen bonding, it's still two bonds just as strong as an A and a T. The G's and C's are the exact same hydrogen bonding. So there's no difference in strength between DNA and DNA-RNA. So there has to be something actively breaking the hydrogen bonds to get the RNA off, so then the DNA comes back together. But we don't talk about it because, you know, it's just one of those proteins in that big complex. Yeah. Yeah, so the quiz, that was just like what we did for replication with the primer. It was an RNA primer. And you guys all told me if there's a T on the DNA and you're adding an RNA primer, what do you stick there? An A, T and A. If there's an A, what do you put there? A U. If there's a G, you put a C. If there's a C, you put a G. That's a DNA-RNA hybrid. So same exact thing for this. If there's an A in the DNA strand, the complement to A of the ribonucleotide is U. The complement to T is always A, either way. The complement to G is always C. The complement to C is always G. We know that it's not the same GC as DNA because the RNA G is a ribose. It's not the same nucleotide, it's just the same abbreviation. It's the same base. The G part of the base is exactly the same whether it's a deoxyribonucleotide or a ribonucleotide. It's just in deoxy, it's attached to a deoxyribose. In RNA, right, it's attached to a ribose. So every single nucleotide in RNA is different than DNA nucleotides, but we abbreviate them the same because it's the base, the actual base on the sugar is exactly the same. So hydrogen bonding between those is essentially exactly the same. Same strength, same setup, right? That's chapter 4. If you go back and look at those stupid structures, right, they fit together exactly the same way. Okay, and what else did we notice at the beginning? We didn't need to have a primer for transcription, and that they call this polymerase RNA polymerase, which makes sense because we're making RNA. DNA polymerase makes more DNA, complementary base pairs deoxyribonucleotides. RNA polymerase, complementary base pairs, and makes phosphodiester bonds between ribonucleotides. And it doesn't need a primer to start, so it can hop right on that naked DNA, that template, and start doing complementary base pairing. Okay? So it's targeted to that spot differently. Right? The DNA polymerase is targeted to where it begins by that RNA primer's 3' OH hanging off. In transcription, it's targeted by the complex sitting on the promoter. They talk about there's the promoter region, then the coding region, and then the termination region. And the termination is totally, that was totally wrong, but we'll get to that as we go. Okay. So how does it know where to start? That's all about sequences, double-stranded DNA sequences in the promoter region. And so when we saw our little picture that in the drawing, right, they sort of set it up as the promoter, the coding, and then the termination sequence. Okay? The promoter is just sequences in double-stranded DNA. Right? And so obviously then the other strand's got to look something like this. Right? It's just sequence, and that's what the initiation factors for transcription recognize, sit there, and they pull in RNA polymerase and sort of set him up to start transcription at the +1 of transcription. So the start site, transcription start site. And if we think about, well, how does that happen? Okay, in eukaryotes, there's a protein that sits here, and then there's another 1,000 proteins. They all sit here. and then they pull in the RNA polymerase. We'll call him RNA polymerase. He's a big fat guy, and wherever his butt sits at the very front is where its complementary base pairing starts. So that's the active site. I can't spell complement base pairing. Right? So if we took the sequence that all those proteins recognize and move it 100 base pairs upstream, we're going to do some, molecular genetics, little genetic engineering, we're going to mess around. If we move those sequences, that's where that protein complex and RNA polymerase will sit, and transcription will start 100 base pairs before it's supposed to. Okay? So it's not that +1 start that controls where it begins. It's the sequences and the promoter that controls it. If we move it 100 base pairs downstream, that's where transcription will begin. Proteins will bind there, it'll pull in RNA polymerase, he'll start complementary base pairing wherever his butt sits then. And you'll miss the first 100 base pairs of your RNA. Okay? So the promoter is the sequences of DNA, and the transcription factors are the ones that bind. The proteins bind to pull in RNA polymerase to start it. So if you're asked what is a promoter, it's DNA sequence. Those other proteins and RNA polymerase recognize to set up the start of transcription. Okay? And so then the coding region is where, right, we'd actually do the complementary base pairing off of one of the strands. Not both, because RNA is only single-stranded, so we only need one of those strands. Again, we're calling this a polymerase, so he obeys the rules of polymerases where they only add to 3' ends. Okay? RNA polymerase is linking together ribonucleotides, 3' OH, 5' phosphate, can only make this kind of thing. Does not require a primer, makes the exact same phosphodiester bond between ribonucleotides as between deoxyribonucleotides. Right? Again, go back to chapter 4 and you look at two deoxys together, the same exact bond as two ribos together. Same exact bond as in replication where we have the ribo and a deoxy, right, from the primer to the first, right? That phosphodiester bond, condensation reaction, same exact thing. Covalent bond. Right? The RNA molecule is complementary to the gene, the template strand. Does that make sense? Which automatically means so we're going to unwrap the double helix because it's just easier. Right? So we're going to say these are the two strands of DNA, and we're just going to pretend for kicks that we're going to make the +1 right there, and we're going to make an RNA that's complementary. Right? So if we had some A's here, what would we have down here? Yeah. Right, we'll just throw a few in there just because we can. And so on and so forth. Okay? What does that mean for this guy here? What would we have had down here? We would have had, if that was an A up there, we would have had a T, 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 A, C, G, C. What do we notice about the RNA and the non-template strand of the DNA? They're the same, except the DNA has T's and the RNA has U's. Right? They have to be. Anything that's complementary to the same strand, they're both complementary to this guy, which means they have to be exactly the same, except the RNA's ribonucleotides and the DNA's deoxyribonucleotides. Right? There's no other way for something to be complementary to the same thing and then be different by the rules of base pairing. So we would call this one the template strand, and we would call this one the non-template strand. It was easier in replication because they were both template strands, right? We wanted to make both. So we didn't have to differentiate which one's template, which one's non-template, boo. But we have to here because we only use one of the strands. We don't use both because RNA is single-stranded. If we made both, let's say some crazy shit happened, and we made both strands, those two RNAs would complementary base pair on each other. And then we'd have double-stranded RNA, which does not work for translation, and boom, done, you'll never make a protein. Right? Because hydrogen bonding happens if two nucleotides that can do it are close enough together. It's just chemistry. Okay? Hydrogen bonding is going to happen if there's anything complementary, DNA, RNA, 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 DNA, DNA, doesn't matter. Right? Anti-parallel, complementary. So we say the RNA looks just like the non-template, except it's ribonucleotides instead of deoxyribonucleotides. Okay? We dig it? We like that? It's your favorite thing ever? It's my favorite thing ever. I'm going to start frothing soon. Just look out. Watch out for the front row. It's like Stein and her vesicles.
Is there a general reason or rule which one becomes template and non-template?
Yes, there is. So exciting. Yes. There's one that's the template, and for any given gene, it's always the same strand. Okay? Once we get through the process of initiation, elongation, all that BS, at the end we're going to talk about how do you know which one's the template. And it's all about it has to make a functional RNA in order to be the template strand. Right? And so every single gene has a template strand and a non-template strand. And it can be top or bottom, it doesn't matter. So even if, on one chromosome, genes can be going this way or this way. It doesn't matter. So if one's going this way and it's using the top strand as the template, if it's going that way, it has to be using the bottom strand. Because again, has to go, if we're saying, I'll bring that up now, this was the +1 and this was the very first ribonucleotide added, by definition, the 5' end of that sucker with its phosphate hanging off has to be that guy. Right, phosphodiester bond here, 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 blah, blah. We'll pretend like there's other nucleotides here. That means this is the 3' OH hanging off, the very last one. Because polymerases can only add to 3' ends, synthesis goes 5' to 3' of the new strand. That, again, what does that tell you about our template strand's polarity? Has to be. And what does that tell us about our non-template strand's polarity? Has to be. So the RNA is not only the same base pairing, except using T, it's the same polarity as the non-template. Because it needs to be anti-parallel to the template, and they're both complementary to the template, so they both have to have the same polarity. And so if you get one of those cheesy kind of quiz questions, just draw it. Don't try to memorize, okay, wait a minute, non-template, wait a minute, is it the same, or is it right? You just draw a little picture, just like we did, right? Just go like this, okay, if she's telling me bottom is template, then here's the RNA, and the RNA, she said left is blue, if you're given that, then you just fill it in. Except not like that, then you'll be wrong. Like Malone did it in class. Right, so you can always draw it out and think it through. The template's the one that's complementary. Oh my God, people! I've lost my mind. Sorry, because I was looking at the red guy. Don't do that! See, I did that on purpose. No, had to go to a PTA meeting this morning, people. My brain's fried. Yeah. Crazy PTA women. Okay. Had to be pleasant and, yeah, sociable. It was difficult for me. Work. Okay. All right, so we're good with that. We're going to keep going over, I'm going to say that a billion times, just like I talk about anti-parallel, anti-3' ends, and you're dreaming about that, and you have nightmares about 3' OHs and all that stuff. So we're going to do, first, we're going to do transcription in bacteria, or PROCs, as I like to call them. Okay? And for 107, it's going to be simplified from, what you're going to get in genetics and the next level up. Because, again, we have to, and then you don't have a billion details. You get the basics, and then we'll add to it. Okay? So for 107, initiation is the main difference between transcription and PROCs and Euc. Elongation is the same, and elongation ends up being the same even at a higher level, so that one doesn't change. Termination, we're going to pretend is exactly the same. It's pretty much the same idea. It's just a little bit different proteins, blah, blah, blah. But I'm not going to totally lie to you, but I'm going to omit some truths, okay, for termination, for simplicity. So for 107, we're going to say elongation's the same, termination's the same, right? But initiation is different for both. Okay, it's different in PROCs, it's different in Euc. And it's too different for me to pretend like they're the same. Like, that would be a disservice, even at this level, for me to generalize it so that it's like that stupid video that said polymerase binds to the promoter. No, polymerase doesn't actually bind to the promoter. Other proteins do that bring in polymerase, which then binds and blah, blah, blah. And it's different how that's set up. But I'm going to try to simplify it, for you guys, but not lie to you. Okay? So, right, blah, 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 that's true. Let's see what else is lying. We already said this, one strand is the template. Of course, everything has to have a billion names, so if you come across other terms, which I will try to always in the quizzes and definitely on the exams, use my terminology, template, non-template. There are books and there are people who use the terms antisense and sense strands. And that's just because if we go back to where our RNA guy is, the RNA we always refer to as sense. Like, it makes sense. That's what we read to make a protein. Right? Translation reads the mRNA, puts the amino acids in order, that makes our protein. So the RNA is sense. It's in the right order. Which means in order to be anti-parallel to the template strand, that makes the template strand antisense. The opposite of sense, anti-parallel to the sense, which is the RNA. I just call it template, because it is. And so then that makes this the non-template, like I call it, or the sense strand of DNA for some people. Because it's the same, right? Same polarity as the RNA, same base pairs, except for GNU. So if there's, if you Google for quizzes and look up stuff online and those terms, that's all that means. Okay? I will use template, non-template, whenever possible. Okay? Because I think sense and antisense is more confusing. And again, I'm not about semantics and memorizing. But again, I don't know what standardized tests pop up what terms. So it's best to at least have an idea. And again, you can work through that. You know the RNA makes sense to be read that way. Something that's anti-parallel to it has to be antisense. Something that is the same polarity has to be sense. So you can think that through without, like, just rote memorizing and then panicking about which one's which. Okay, and then this is just showing us sort of what that little video did, and so they're pointing out template and non-template. And then I'd also point out that if that's where the RNA started, this region up here is the promoter. The sequence, the DNA sequence just upstream, just 5' when we're talking about the actual strand of RNA. Upstream, 5' of the RNA, right? That's the promoter, because remember I said that's where RNA polymerase had to have originally sat down to start complementary base pairing right there on that guy. And we know if it's labeled already 5' to 3', even if they didn't draw an arrow, if I gave you something, write an arrow which way it goes, just like we did for replication. We know it has to proceed in the 3' direction. Okay? So in this case, there's only one template strand. And so that RNA is always, if we want to think about relating it to replication, to the crotch, it's the same idea, the strands following the crotch, right? And so in reality, the polymerase guy is now here, and within that complex, you know, there's 50,000 proteins around here. Somebody's job is breaking the hydrogen bonds there, too. So we don't name it, right? Well, the one that breaks hydrogen bonds in replication is helicase. We just don't name it. It's part of the complex. That's nice and easy. We don't have to worry about it, okay? But somebody has to do it because it's not just a spontaneous, woo, right? And then if we look at what's actually happening, right, we can look at, notice the yellow has an extra oxygen at the 2', right? They don't even show it on the DNA, right? So ribonucleotides, the bases are exactly the same. Hydrogen bonding is the same between U and A as T and A, the same kind of strength, right? Phosphodiester bonds along the strand, hydrogen bonds between, right? Brings its own energy when it cleaves those phosphates off to do that phosphodiester bonding, right? Condensation reaction, lose water, same idea. Okay. Oh, yes, sir. You betcha. Yes. Yeah, so the polymerase is traveling, the whole complex is traveling in complementary base pairing. So again, it's like they took a snapshot of that picture, and it's not exactly right because really these two guys would have already come back together. But they're showing it open just so you could see that you know, the new strand of RNA is in there. If we took another snapshot once it's open even farther, right, if we did this and now we're down here, oh, let me see, do I have a yellow we can actually see? Right, the RNA would then be coming off already. Right, just like in that little video it showed it, and here's the 5' end. I can't freaking read that. Right, I'll go back to green. You get the idea. This is the way it's going, that's the 3' end. Okay, and this, if it had started down here, this is our, you know, this guy came back together already. And so, yes, literally, it's dangling off. It's held on by the, however, I don't even know, 20, 30 that are the DNA-RNA hybrid as it goes, but the rest is already dangling right off. Okay. And in prokaryotes that don't have a nucleus and everything's just in the translation can start immediately upon, as soon as enough of that RNA comes off, a ribosome, small subunit can grab that puppy and assemble and start doing translation. In Euks we can't because we're still in the nucleus and the ribosomes are out in the cytoplasm. But in Prokes we can. That makes sense? And again, it's this ongoing process. We're just showing you pictures of, you know, it's not like waiting for 20 to go and then the fork moves and then waiting and then, you know, it's all just happening. Okay. So let's do a little bit of polymerase structure just because this helps us with initiation. So, and all of these things come back to 50 years ago, or whenever the biochemists isolated these enzymatic properties, they named it RNA polymerase. And then as they got more specific and better assays, they realized, oh, this isn't one protein. Now we know it's actually 2. And then it's 3. And then by now it's 50. Right? There's just so many different, so it's, but it only works all together, so that would be the, massive quaternary structure. Right? Not just 2 polypeptides together, 50, or some ridiculous number. For us, the most important ones, and they call the whole thing, they love this word, so this is a total standardized test word, holoenzyme, meaning everything together. This has to be together for it to function. Okay? And so for initiation, one polypeptide is called the sigma factor, and then the rest of it, which they used to think was 1 and now is more than that, but we're just going to pretend, the rest of the core enzyme that has the complementary base pairing phosphodiester bonding function, active site. The sigma factor's job is to bind to the DNA sequence in the promoter. Okay? So this one actually binds the DNA. This one has the enzymatic function, right? So the active site. So without the sigma factor, the core enzyme can't do anything because it has no way to target itself to the start of transcription, to the promoter. Okay, so we need sigma factor. Sigma factor binds first to the sequences of DNA, and then you can think of RNA polymerase hopping on its back to set itself up to start complementary base pairing. So wherever the sigma factor binds, that controls where transcription starts because it pulls in RNA polymerase, or the rest of the core enzyme all together, the holoenzyme. Ooh, it's so exciting. Okay, so that's exactly what I said in another way. So we'll just go there. And so if we go back to my little stupid drawing, and this is the promoter. Right, we'd have sigma factor that binds, then RNA polymerase, right, and then that's how it sets up. Right, and then we'd break it open and we'd start making the RNA. Okay, so sigma factor's job is to bind DNA and bring in RNA polymerase. And so it's a DNA binding protein. Its job is to bind to DNA. And so here's a little picture of how that works. And again, it's not just any random sequence of DNA. Otherwise, sigma would sit down all over a bacterial chromosome and randomly set up transcription. Right? Transcription is very tightly controlled so that we're only making, right, the exact RNA that we want to. So essentially, I'm kind of lying, but lots of them, most of them, have a specific sequences in this promoter region, that region right upstream of where transcription starts, that sigma recognizes. And we're not going to memorize the sequences, because that's stupid. Again, right, we don't care. If you guys end up studying the sigma factor in graduate school, you'll remember those sequences. But that's just a dumb multiple choice question to memorize a consensus sequence. I want you to know that they, what did they used to call it? I don't know. I'll look it up because I think they still call it something else on standardized tests. Yeah, so this right here, hang on, I think, 1, 2, 3, 4, yeah. So this would be the +1 of transcription. So this whole region here, right, is the promoter. So what they're not showing us, right, is just the rest of that strand of DNA. And then they're not showing us the other strand, right? The promoter is double-stranded. They're just showing us the sequence of the one strand for simplicity, but really, in the promoter region, it's always together. That's not where it opens. It's always together. It only opens where we're going to start complementary base pairing. But this minus 35 sequence and minus 10 sequence is what the sigma factor is looking for in that specific distance apart. So if we move the minus 35, and if you count it, it's just this is plus 1, minus 2, minus 3, minus 4, minus 5, going in this direction. So if you kept counting, right, the DNA keeps going, minus 100 would be over there, right? So we count backwards in the promoter regions. That's how you can tell it's not coding for a protein if it's got a minus number, right? Whereas the RNA, we call the first base pair of RNA plus 1, the second plus 2, plus 3, plus 4, plus 30, plus 500, however big that RNA is. So the sigma factor, they're showing this guy, right, sigma is blue, this blue blob right there, is contacting DNA at both that site and the minus 10. It grabs it and pulls in RNA polymerase to start transcription. So that's transcription initiation, essentially. So I want you guys to know that sigma factor requires this minus 10, minus 35 to pull in RNA polymerase to start transcription. The minus 10, minus 35 sequences are part of the promoter. And they're really double-stranded, but they're shown single-stranded just for simplicity. But you can see, right, this guy here, right, they show it double-stranded when it's actually binding. They're just zooming in on one of the strands just for simplicity. Okay, so in prokaryotic transcription initiation, sigma binds to the promoter, specifically at the minus 10, minus 35, brings in RNA polymerase. RNA polymerase is the one that starts doing the complementary base pairing and makes the phosphodiester bonds. Okay, see him? Zoom in, yes, yes. So sigma's following at the beginning and helps open up the helix. We start complementary base pairing. Again, remember that this stupid little abbreviation here just means ribonucleotides. Any of them, N stands for A, G, C, or U, right? Nucleotide triphosphate, so any of those. If it were, we could call them for DNA replication, we'd call them DNTPs. So deoxyribonucleotide triphosphate. And so that would be where initiation begins, transcription begins when we start complementary base pairing, incorporating those NTPs into this chain of RNA, phosphodiester bond, complementary to the template strand. As soon as initiation is essentially complete, the sigma factor goes away. So we don't need sigma for elongation, we just need him for initiation. So now something else has to be opening the helix, because again, hydrogen bonds don't just disappear, but we don't care what it is. Other proteins in the complex. Okay? And so we're just and see how the RNA is falling off, right? It's not stuck completely to the template, and it's nice because they're showing this coming back together, right? So the DNA comes right back together behind it. If we would draw an arrow, right, it's going in that direction. This was the beginning, so this had to be the 5' end, right? The guy that's flapping off was the very first one, which means down here is the 3' end. And so that's all they're saying. Downstream, it's going that this is the upstream region. So wherever this aren't, like if it was that was the beginning, then up here would have been where the promoter was. Okay. And so then elongation just happens. It keeps going, it keeps going, it keeps going. Unlike the big fat lie in the video, termination does not happen the way they said. Okay, and Termination of transcription is all about sequences in the RNA, which are controlled by sequences in the DNA, but it's not like a stop codon in translation. If we remember back from whenever we did translation in high school, right, when the ribosome comes to a stop codon, it blows apart, there's no amino acid, everything falls to crap. Not how it works for transcription. The RNA polymerase transcribes right through the termination sequence as if it doesn't know any better. And it doesn't, as it turns out. So it transcribes the RNA all the way through, doesn't care that it's a termination signal, doesn't know that it's a termination signal, would keep transcribing down through the entire chromosome if we let it. Would never stop. No sequence stops RNA polymerase. It just keeps going and going and going. What happens is the RNA that comes off from the termination signal is complementary to each other. And the RNA actually hydrogen bonds to itself. That's recognized by a complex, which kicks the RNA polymerase off the template. Okay? And so that's what this is talking about. Transcription ends with termination phase. Transcribes through this signal, which is a sequence of RNA that forms a hairpin, that disrupts everything. Well, that's not that. Oh, hang on. Wait a minute. Where's my blank? Oh, people. Oh, people. Okay, let's do a blank. So what I mean by that is, and I'm going to try to do this a little bit stepwise, even though it's going to be a stupid-looking picture. So I'm going to start from here. We're going to say this is the promoter, and then here's the rest of the gene. Here's what we'd call the term sequence, and then this is, whatever else. This is what they called it. They called it coding, right? That's really nice. Okay, so what happens? We'll say -10 and -35 sequences are in the promoter. Sigma factor, our good friend, binds to those. And let's get another color. Let's get this guy. RNA polymerase comes in. Right, the blue guy. And we get the start of transcription. Let's try purple. I think we can see. Okay. We'll put one nucleotide there. Okay. If we take another snapshot in time, the purple's the RNA nucleotide. 1. Okay. I'm not going to draw polymerase in sigma because that'll interfere with all the other stuff, but it's moving, right? Or actually, I'll do the RNA first, and then maybe I'll put it on top. Okay? And so now we'll say this is the very beginning. We've got a tiny bit. This is the 5' end and the 3' end, and it's moving in that direction, right? So if we're still calling it initiation, the complex has the sigma factor and RNA polymerase, right, and it's adding the nucleotide. Okay, we're going to take another snapshot in time. We jumped a little bit farther forward. Right, now we know polymerase is still right there. Sigma has gone away. We don't need blue, we don't need that color anymore. Polymerase is right there, right? It's still doing transcription. Now we're in the middle of elongation. And it's just going to keep transcribing, keep transcribing, keep transcribing. And since I drew too big, I got to move this guy over a little. Okay, so now we're going to take another snapshot. Oops. transcribing. Okay? This obscene-looking structure, inappropriately drawn here, is what they call the hairpin. Let's do another color. So that's what that other slide called the hairpin structure. That's really like if we want to zoom in on it, and I'm actually going to draw, we'll just zoom in, right? That would be like, right, remember the line is just nucleotides, and we'll make this a G. It's just a strand of RNA closing on itself because there's a string of A's and a string of U's close to each other, and whenever nucleotides that are complementary are anywhere near each other, hydrogen bonding happens. Yeah. That would be nice if they just called it a stem loop, just like we talked about for tRNA, but they save that for tRNA and they like to call it a hairpin. So like, yeah. Like a hairpin turn, right? Nobody wears hairpins anymore, but like a hairpin turn going up to Big Bear, you know, you're just winding up the, right? And so it's just, yeah, it's like a stem loop or a hairpin. It's just complementary base. It's double-stranded RNA, except it's from a single strand. It's just closing. It's not two separate strands, right? But they've complementary base paired to each other. So they call it the termination. You know, we call it the termination signal up here in the DNA because that's where, the only way you can get a string of A's next to a string of U's is if the template strand had a string of T's and a string of A's. Right? So up here there was a string of T's, and let's see, and then a string of A's. So when we complementary base paired, we got this string of A's and string of U's. Boom, they close together. So that would, exactly, that would be secondary structure of that RNA. And so really, we always think about RNA as a long linear piece, but if there's stuff that's complementary, it will fold up, just like a tRNA. tRNAs are built with all this complementary shit, so they do fold up into that secondary structure. And so yeah, that's a secondary structure in the RNA, and as it turns out, let's find a different color. I don't have enough good colors. How about this one? A protein complex recognizes this, bunch of proteins, and then they essentially, I don't know if I can do this. That's supposed to be a shoe. Kick the crap out of RNA polymerase and it lets go of the DNA. So kind of like what happens at the end of translation. To stop codon, a release factor comes in, there's no tRNA there, and it explodes. A miracle happens, and the ribosome falls apart. Right? This protein complex recognizes the hairpin. It's still, right, it's close enough to where it's attached at the DNA, where our good friend, you know, RNA polymerase is sitting, that it essentially causes that complex to fall apart. So if we mutated the termination signal and we don't make a hairpin, transcription would go until it ran through something that made a hairpin to get this complex to kick it off, or until it fell off the end of the chromosome. So it's the sequence of the RNA that actually causes termination. The sequence of the RNA is dependent on the sequence of the DNA because that's how it exists. So they call it a termination signal in the DNA, but the DNA sequence doesn't do shit all by itself. It has to be transcribed into the RNA, the hairpin or the stem loop, the protein complex recognizing, booting off RNA polymerase. That's termination. Same idea for Prox and Euks, it's just a little bit different protein complexes that do it. They know more details than just, I told you there's a whole bunch of proteins. But essentially the very same idea. Same kind of hairpin, same kind of kicking them out at the end. Okay. That make sense? You dig it? So when the question comes up, RNA polymerase going, as it transcribes through the termination signal, don't fall for, and then it falls off. When RNA polymerase recognizes a sequence, it falls off. No. Think of RNA polymerase as really dumb. Oh, I'm just going to keep going until somebody kicks my ass, and then I fall off. So the RNA polymerase doesn't recognize squat for termination. The protein complex that recognizes that hairpin, that secondary structure in the RNA that causes termination. Okay, so we're just going to do a little upload since we haven't done any. So we're going to pretend like this is part of a gene. We're going to go ... so we know this isn't like an entire chromosome or anything crazy like that. And if I tell you, which I'm going to tell you, we're going to make it easy. I'm going to tell you the bottom strand is the template, okay? And I'm going to tell you I'm going to tell you that here, going this way is the promoter. I want you to give me the sequence of the RNA that comes off this guy. Okay? So write out or type out, however you want to do it, the RNA. Complementary base pair, and then you can just do ... at the end, because obviously we're not going to get to a termination signal. You're not going to need to make a hairpin or any of that crap. Pretend like we're just the beginning at initiation and the beginning of elongation. You don't have to draw any proteins, but you know that sigma, our good friend, sigma would be up here somewhere at -10 and -35. And you pull in RNA polymerase, and we'd start, at the first base pair past what's called the promoter. And so if the bottom strand's a template, that makes the top strand the non-template. And then screenshot and upload to whatever the first upload is of the day. Are you trying to type it? Yeah, I know. And they're like, why don't you just type it? I'm like, because I suck at typing. And neatness doesn't matter for this. Because you are grading your own, checking your own for after I fill it in. So it doesn't matter how messy it is. Oh, and sorry, because I forgot to tell you. So these pictures that are not in your PDF, because I didn't get my shit together early enough, are linked in Moodle. So if you want to upload it into your Explain Everything, it's in the Moodle. It's just like a JPEG image or something. So if you don't want to type all that out. Or you can just do the RNA. You don't have to do, you don't have to write what's up there. But if you wanted, there's the other ones I'm going to use today and to finish tomorrow are all linked in there because I didn't get it all together in time. You don't have to. Just know that it's, I'm just reminding us, how I have to repeat everything 1,000 times. Yeah. Right, yeah. Right, because if that's the promoter, we don't transcribe, that's a good point, we don't transcribe the actual promoter sequence, right? We start at, like if I was going to draw our big fat polymerase, this will help help you guys. I give your blue, right? The promoter is, right, polymerase would bind on top of sigma like that, and he wants to start, anything that's actually classified as promoter is not transcribed. So again, tricky questions like, what parts of a eukaryotic gene are transcribed? If it includes promoter, no. What do we transcribe? Exons, introns, which we didn't get to. Termination signals, are those transcribed? Yes. Right? Because it has to make the stupid hairpin. How do you make a hairpin if you didn't transcribe it? But the promoter is never transcribed. Right. Yeah, so anything upstream, anything this way is the promoter. That whole side is all promoter. Yeah. Well, the promoter doesn't move. Right, no. Yeah, so polymerase, here I'll draw in blue. Yeah, polymerase guy wants to go this way. I wasn't really telling you I was going to get us to there. The green thing is the sigma factor that's bound to the promoter. Sorry, it says, you can't read that? It says minus 10, minus 35, because really you're counting backwards from here. We can't really see where it binds because this is minus 1, minus 2, minus 3, minus 4, minus 5, 6, 7, 8, 9, 10. Oh, crap. Stop that. Stop that. Stop it. What did I just do? Oh. No, I don't want to do that. Okay. So then the RNA polymerase, yeah, travels down, finds the sigma factor on the promoter, and then starts traveling. Right, breaking hydrogen bonds to get a single-stranded template and complementary base pairing to the template strand, right? Yep, just like that. Just like that, yeah. The termination on the template would look like a string of T's and a string of A's. To get a string of A's and a string of U's. You know, I don't even know. Not a million, but probably 10 to 15 of each. Yeah, because it would have to be because randomly you might have 3 U's and 3 A's, and you don't want that to terminate. So yeah, it makes a good. That wouldn't randomly happen. Yeah. So I'd say 10 to 15 in a row, and then a couple on the loop. You know, I just drew one, but and then more. All right. So we're using the template strands. And so this would be our first base pair on the template strand that we want to complement to. So what do we have here? What would be our first ribonucleotide? U. And this is the 5' phosphate hanging off, right? His 3' OH is sitting there waiting for the next one to come in. Right, so then what do we get? We get U. Oh, I'm erasing. Supposed to be making my oh crap, now I don't know where I am. Oh, am I here? No. What did I do? Help me! Oh, I did it wrong, that's why. Yeah, I'm like, what? Where am I? A, G, U. Oh, I heard a T! I almost heard a T. And then dot, 3' N. Right? You dig? You're with me? And for this, we don't give a crap about the non-template strand, right? Whatever, it's just sitting there. We don't use it. We don't need it. We're not going to care about it right now. We might care about it. We're going to care about it a little bit later. Okay. Yes? Yeah, either place. It doesn't matter. Yeah. And then again, we notice it's the same polarity as the non-template and the same sequence, except it's ribo instead of deoxyribo. All right, let's move on. Okay, so let's do Euks real quick, and it's really just initiation that's a little bit different. Same idea, though. And there's, again, just like when we talked about replication and the DNA polymerases, there's 50 billion DNA polymerases in Euks. There's lots of RNA polymerases in Euks, and they all have a different function, and some of them are redundant. We're not going to care about any of them except for RNA polymerase 2, and I'm probably just going to call it RNA polymerase, but I mean RNA polymerase 2. That's the one that's in charge of transcribing mRNA. And for us, that's all we're talking about for transcription is making mRNA. We know in order to make tRNA, you have to do transcription, because that's the only way RNAs just don't appear out of nowhere, right? That's a different polymerase, and we're not going to worry about, but it's the same idea, right? And then all the other crazy RNAs that do stuff, we're not going to talk about in 107 either. Okay, but rRNA, right, has to be transcribed. tRNA has to be transcribed. MicroRNAs have to be transcribed. But we're going to focus on making mRNA because we're trying to get to proteins, right? That's our point right now. But we know all those exist, and we love them, we just don't have time for them. So in eukaryotes, there's nothing called a sigma factor. There's an analogous protein. Analogous meaning it's different, but it does the same thing, right? Like an analogy. Tall is to short as... No, we don't want to think about analogies. So no sigma factor. The protein that binds to the promoter, that actually recognizes a sequence in the promoter of eukaryotes, is called the TATA binding protein, or TBP. Okay, in lots of eukaryotic promoters, that's our sort of example of how it works. It's then bound by a whole bunch of other proteins and then pulls in RNA polymerase. So same idea as prokaryotes, but more proteins involved and a little more complicated. But a protein binds a sequence in the promoter. It's actually at the -27. Okay. Whereas, right, if you hear -10, -35, prokaryote. Sigma factor, prokaryote. If you hear -27, or the TATA sequence, or TATA binding protein, eukaryote. Okay? Transcription factors are all the other proteins that help hop on top of the TBP, the TATA binding protein, bring in RNA polymerase to start transcription. So TF2D is the one that our book, I think, still talks about. Again, this was, right, originally it was RNA polymerase. And then they got more sensitive, and ooh, we found 5 things. Let's name them transcription factor 2 for RNA polymerase 2, A through F. And then they found some more, okay, G through M, and now they're past Z, and each one of those is a set of 10 or 15 proteins, not one. So it got all crazy, but the protein that binds the sequence on the promoter is called the TATA binding protein. And it's conveniently called that because this is the actual sequence in the DNA. Woohoo! So you don't have to remember. You could memorize the consensus sequence for that because it's the same as the name of the protein, but we don't need to. And it's at about -27. Okay, so that's in Euc. So if we look at the little picture, part of TF2D is the TATA binding protein. Right? They're just showing the whole thing. So that's actually the one that recognizes this TA, and then a bunch of A's sequence, assembles all the rest of the crap, and brings in our good friend RNA polymerase, right? These guys are in charge of taking care of the helix. This guy actually sits closer to the start to make sure the active site of polymerase 2 is right there. Well, a whole bunch of crap. All I want you to know is TBP and RNA polymerase. Okay? So this is the guy that actually binds the sequence. And so if we were counting backwards, if they're saying that red arrow is where transcription starts, that's the +1. -27 is right down here at that TATA sequence. Shit, we're running out of time, people. The hell. Okay. What else do we got? What else we got? What else we got? We got this, we got that, okay. So that's eukaryotic initiation. Elongation's the same, termination's the same for us. Okay, same hairpin type thing, proteins recognize it, kicks off RNA polymerase. Okay, so the main difference is that initiation. Tau binding protein brings in RNA polymerase for eucs, sigma factor brings in RNA polymerase for procs. Okay. So in prokaryotes, that's it. We do transcription, we get an mRNA, as soon as it starts coming off, like I showed in that little picture, you know, the dangly guy at 5' end, ribosomes can grab onto it and start translation, because there's no compartmentalization, right? The mRNA comes right off, it's ready to be translated. In eukaryotes, it's not true. First of all, we're stuck in the nucleus. First, we have to get out of the nucleus before we can do translation. And second of all, we have junk DNA, which really isn't junk, but that's what they used to call it, within our RNA that we don't want to send to the cytoplasm for translation. We have non-coding sequences. Okay. Within eukaryotes, there's parts of each gene that do not code for protein. They're not a triplet. You don't want to send them to translation. We have to remove them first. So coding sequences are called exons. We keep those. The crap that we don't want, that we want to cut out, are called introns. So if I were going to draw a picture again, back to our original stupid drawing, right, we went promoter, coding, termination, sequence, you know, end, blah, blah, blah. Within this coding region, we have exons and introns, okay? And these are the ones that are crap, that we don't want. Okay? Now remember, RNA polymerase is stupid, doesn't know diddly, can't recognize a termination sequence, can't recognize an intron if it smacked it in the face. And so when we do, there's our, oh crap. So when we make an RNA, right, we're going to start here. Well, I guess I'll draw it down here. And we make the whole thing, right? Through the termination sequence, I'll add our little hairpin there, right? And we include both exons and introns in our RNA. We do not call this an mRNA. This here is either called pre-mRNA, I think that's what our book calls it, or all the genetics books call it a primary transcript RNA. Not ready for translation because we have to get rid of the introns first. Okay? So again, when asked what does transcription do, does not transcribe the promoter, yes transcribes exons, yes transcribes introns, yes transcribes the termination sequence. Right? If you're asked about prokaryotes, does not transcribe the promoter, only has exons, so don't say introns, they don't even exist, right, and transcribes the termination sequence. Okay? And so before we can turn this pre-mRNA or this primary transcript loose into the cytoplasm for translation, we have to process it. We have to do stuff to it to make it ready for translation. We put a 5' cap on the 5' end, that's why it's called 5'. We put a whole bunch of